Hello, this is the second lecture in the first unit, and we're going to be talking about the scientific method today. You're going to be designing experiments at the end of this unit, and you're going to use the basic outline of the scientific method, which goes like this. First, it's based on logic. It's really a way of thinking rather than a standardized set of procedures. Many times, but not always, it begins with observing a problem. For instance, we could look at, say, energy drinks. Does teenagers drinking energy drinks, does that cause any problems? We could ask a question and formulate a hypothesis saying, increased use of energy drinks can cause increased heart problems. That would be a hypothesis that we could test. And then we would design a controlled experiment. Now, of course, we couldn't really do this as much with people because of ethical dilemmas, but we could follow two groups of people, one that drank a lot of energy drinks and one that didn't, and we could examine their medical records for heart problems. And then finally, it's very important for a scientist to share results. There is a huge problem in the scientific community with experiments not being reproducible. And this means that the same setup will give you different results. And of course, we want experiments to be able to be reproduced because this shows that the conclusions are probably correct. Now, we're going to talk about this in terms of spontaneous generation. This is a very old concept. You can see on the left, there's a picture of maggots. Maggots are one of the creatures that people believed came out of nowhere. And this is what spontaneous generation means, that life could appear from non-living matter. For instance, maggots just appear on meat. The meat physically makes the maggots. Now, this was not seen as something that needed to be investigated, but of course, as the scientific minds started to approach different problems, they looked at a lot of things that people thought were common sense that turned out not to be. And this guy is a name called Francisco Redi, and he proposed that maggots don't come from the meat, they come from flies. And this was a pretty new approach, and a lot of people thought that that was crazy. And so what he did is he tested his hypothesis that maggots come from flies by placing some meat in jars and then covering half of them. When he did this, he was able to see whether or not the meat that was covered developed maggots. And in this experiment, everything was controlled, which means kept the same, except for a single variable that he was testing, and that was the covering on top of the jars. It's really important when you design your own experiment that you keep everything the same, except for the single thing that you are testing. This means that your results can be attributed usually to that single thing that you changed. So this is a picture of what he did. In his uncovered jars with meat, the flies were able to land on the meat. In the covered jars, they were not. And in the uncovered jars, you see that maggots appeared, but no maggots appeared for the covered jars. Now, just a brief little sideline here, types of variables, so that we can look at some of the other experiments and know what the different variables are. We tend to call the variable that is being changed or manipulated the independent variable. In the case of Redi's experiment, that was the jar covering, the single thing that he changed. And the dependent variable is a little bit harder to understand. It's the one that changes in response. So sometimes it's called the respondent variable, but in this case it was the presence or absence of maggots. Another person named Spallanzani wanted to test a similar experiment or a similar proposal on spontaneous generation because, of course, one experiment proves nothing. You must have thousands of experiments in order to disprove a theory. So he tested Reddy's results, and what he did is he had a flask of gravy that he boiled to kill anything in it. Now, the first flask he left open, and within a few days it became cloudy. So it was very full of bacteria and other microorganisms. In the second one, he boiled the gravy, but sealed the flask and found that it didn't become cloudy. There was no microorganisms at all. So the independent variable here is whether or not it is sealed. So is there air going in? And the dependent variable is going to be whether or not microorganisms actually show up. So again, what we're changing is the independent variable. The thing that we're looking for is the dependent variable. And finally, Pasteur's experiment. You have probably all seen on the side of your milk carton, it says pasteurized, and that was named after Louis Pasteur. He was the inventor of the process to kill most of the bacteria in milk. Because raw milk doesn't last very long. It would last about a day or so in the fridge. But once it has been heated up, it will last weeks. Now, Pasteur provided final proof that spontaneous generation didn't occur, because even after all those experiments, there were still doubters. People said the reason why there was no maggots, there were no bacteria, is that they didn't have enough air. And so he decided to test this. What he did is he invented a very cool Z-shaped neck, this curved neck on top of a flask. And so he boiled broth, which is made from, say, chicken bones and organs, so a great medium for bacteria to grow in. He boiled it, and then he kept it. And it was free from microorganisms for a year. But then the moment he snapped off the curved neck, bacteria got in, and this caused the broth to become very cloudy. Now you can see that because of the curved neck, there was plenty of air getting in, but the bacteria couldn't because they would fall in from the air and be trapped in the neck. So his hypothesis was that the microorganisms weren't just going to appear out of nowhere, that they came from somewhere, in this case, the air. The independent variable, remember that's the thing that he changed, is going to be the curved neck of the flask. And the dependent variable is the thing he was looking for. And just like in the other experiments, it's whether or not there were microorganisms. So all of this led to a new theory. A theory in science is something that has been supported thousands, sometimes hundreds of thousands of times. We don't really like to call them facts because a theory even can be disproved. But this is a very well-supported hypothesis. So when a hypothesis has been verified many different times, it becomes a theory. So all of these scientists' experiments led to the theory of biogenesis. So bio meaning life, and genesis means beginning. So this means that life comes from other life. It doesn't come out of nowhere. And the goal of theories is to explain phenomena or events in nature. You'll often hear things like, oh, it is just, evolution is just a theory. But in science, theory is the highest level of proof that we have. For instance, there is more evidence for evolution than there is for gravity, because we, as of now, science cannot explain why gravity occurs. The last thing we're going to talk about is what actually makes something alive. Because tomorrow you're going to be working with a mealworm, and that's a beetle larva, and you're going to have to figure out whether or not it is alive. So hopefully you know a book isn't alive. A tree, most people would say yes. But then we've got a computer. Most people know a computer isn't alive, but they couldn't really point out why it isn't alive. They'll say things like, oh, well, it can't move, it doesn't have a heart, but that is true of many things that are alive. So we're going to quickly look at the 8 characteristics of life. Now, this is our definition, and of course there are some other definitions, but these are the most accepted. First is that it's made up of cells. We could definitely test that in your mealworm. Take a little bit of the mealworm and look at it under a microscope. It must reproduce in some way, which your computers definitely do not do. It must contain RNA or DNA, which is the universal genetic code. And it must go through growth and development, just like you currently are going through puberty. That is a phase of development. All creatures must take in energy in some way. For instance, plants take in the energy of the sun and use that to make sugar. And then you, as an autotroph or as a heterotroph, eat those plants and you get the energy from them. All creatures must respond to their environment in some fashion. Their responses don't need to be the same, and you'll be testing that with your mealworm. And they must do something called maintain homeostasis. This means keeping everything in the body stable and balanced. In humans, some examples of homeostasis are temperature. Even in a very hot place or a very cold place, your body will maintain 98.6 most of the time. And finally, all creatures must evolve or change over time. This doesn't mean individuals, but it does refer to species. Species don't stay the same. If you look at things over a very long period of time, you are going to see a lot of change over time. Make sure that you have filled in the summary of your Cornell notes and written questions. Those questions are, remember, going to be checked tomorrow when you come into class. Thank you.